Hello, good evening to all of you. Am I audible to all of you? Can you please confirm? Is the screen is visible? Is the voice is audible? Okay, fine. So welcome to the third day of CUET UG Vijeta Series 2022. So as you all know, it is a central university entrance exam or entrance test we can say, which is being conducted by NTA. So mainly these we are going for admissions of UG courses. So BioTecNika has started with the series 2 days back and it will be there till 13th July. So you will come across some of the important topics from your syllabus. I hope so you are enjoying the 2 day session which has occurred recently, and today we are into the third session. So today we are going to discuss about unit 2, that is translation. So shall we start with the session today? Can you please type BIO in your chat box so that we can start with the session? So you are aware of what is translation is all about? I hope so you have studied somewhere what is translation is, right? It is related to what? It is related to protein synthesis, right? So before getting into the details about translation, just wanted to inform you that we are having a Telegram app, and also you can download our BioTecNika app either via Google Play or App Store. So you will get all the notifications, all the job alerts, all the admissions, latest news, everything you will get at single platform, and you will get all the information regarding the life science. Okay, so going to our session. Before starting with translation, can I get some answer that what is meant by central dogma of life? Have you heard of this sentence before? Central dogma of life. Anyone? Yes? Have you heard of it? No? See. Central dogma of life mainly includes what? It includes protein synthesis. But to get this protein, we have to carry over various processes. And those processes includes transcription and translation. So basically, the central dogma of life is mainly related to the flow of genetic information within a cell. So if you can see, we have our genetic material DNA, right? Now this DNA will be coding for RNA. And how we can get RNA out of DNA? With the help of a process known as transcription. Next, what we are having? We are having RNA. So this RNA will be coding for what? RNA will be coding for protein. And how we are going to get this protein? With the help of a process known as translation. So in eukaryotes, specifically speaking, transcription occurs in nucleus and translation occurs in cytoplasm. More precisely speaking, it occurs in ribosomes. So basically, this is a unidirectional flow of information. That means it will move from DNA to RNA to protein. There is no reverse order. Of course, there is a reverse order is there, but that is a different story. It may be that some viruses will be having a specific enzyme that will help in the conversion of RNA to DNA. That is completely different. Today, what we are looking into this unidirectional pathway. That is how DNA is getting converted to RNA. Finally, how RNA leads to the formation of protein synthesis. So, if you look into this picture, this is our genomic DNA strand. And you are aware that within this DNA strand, we are having two regions. One, you call it as a coding region, and another, you call it as a non-coding region. It is your coding region that leads to the production of proteins. So, as you can see, within this DNA, some polymerases will be activated. So with the help of this polymerase, you are getting your messenger RNA. Into this messenger RNA, when ribosomes are recruited, you are getting a nascent protein. And this protein, what happens? It will undergo proper folding and it is ready to be imported to different organelles, because we have got variety of proteins and we have got number of organelles. So depending upon the necessity, the proteins will be added or will be moved to different organelles. Next, so before going into that, what is the basic difference between DNA and RNA? As you can see, the very much strong difference or the basic difference is that DNA is double-stranded helix, right? But when you look into RNA, this is single-stranded. And another difference between DNA and RNA is that you are having 4 nucleotide bases, right? Thymine is there, adenine is there, guanine is there, cytosine is there. So you might be aware that A pairs with T and G pairs with C, right? You might have studied it under replication. So when it comes to RNA, instead of this thymine, we are having uracil. So that is the second important difference between DNA and RNA. So now since you understood the difference between DNA and RNA, let's check out what is exactly a protein synthesis is all about. Protein synthesis mainly comprises of two major factors. First part becomes your transcription and second part becomes your translation. And to carry out these two major processes, we must have RNA, DNA as well as a set of enzymes. So let's look into the processes. So before, I'm just giving you a glimpse of transcription, not going into details, but translation I'll be going to details. So how does transcription occurs? As I said you already, we are having RNA polymerases. So these enzymes will be recruited to a specific region within the DNA. You might be aware that region is called promoter region. So once this polymerase goes and binds to the promoter region of the DNA, what happens? Unwinding occurs. Unwinding in the sense separation occurs. And after that, you can see mRNA synthesis is occurring. So in this case, what happens? The same A will be pairing with T, but since we are having instead of mRNA U, so A will be pairing with U and G will be pairing with C. After that, what happens? Nucleotides will be added unless and until your RNA polymerases reaches a specific sequence known as termination sequence. And at that time, what happens? mRNA will be released from the DNA, as you can see from here. So from here, the mRNA will be released from the nucleus and enters into the cytoplasm, because we know that within the cytoplasm, we are having the ribosomes, and it is in the ribosomes the protein synthesis is carried out. Okay, so this is a glimpse about transcription. Now, here I'm just explaining you shortly. After some time, we'll go into details. Next, what is we are having? We are having translation. So as I said to you, for translation, ribosome is the protein synthesizing machinery. So for initiating a translation, you must have a specific codon. Now regarding codon, I will come into the upcoming slides. Right now, you remember specific sequences. Okay. Within the mRNA, we are having specific sequences known as start codon into which your ribosome will go and bind. After that, what happens? As you can see in this picture, tRNA will be recruited. Then tRNA and mRNA, they will be having some complementary sequences into which they will be bound, as you can see in the third picture. Now the ribosomes will keep on moving and the amino acids will be keep on adding up. Soon when it reaches a termination sequence known as stop codon, what happens? Your ribosome will be releasing the mRNA as well as the amino acid chain. And what happens to this amino acid chain? Amino acid chain is nothing but a formation of proteins. What are proteins? Proteins are made up of different amino acids combined together, leading to a polypeptide. So this amino acids will be provided with proper conformation, resulting in the production of protein. So this is all about your translation. Now, since today we have a topic of translation, we have to look into details about that, right? So the machinery responsible for translating the language of mRNAs into the language of proteins is composed of four major components, right? What are the four major components? We are having mRNAs, tRNAs, amino-acyl tRNA synthesis, and ribosome. So students, please note down these 4 points. I am going to explain you all these 4 points individually. So the first point which I am going to explain you is mRNA. So we have got 3 types of RNAs that are being involved in protein synthesis. What are those RNAs? First one is ribosomal RNA. Ribosomal RNA, as the name indicates, this is the component of ribosomes, meaning these RNAs resides within the ribosomes. Second one is transfer RNA. Transfer RNA, as the name indicates, it is transferring something. So what does it transfer? It transfers amino acids to the corresponding mRNA. And the third yet important RNA when it comes to translation is your messenger RNA. Messenger RNA, as the name indicates, it is having some message. That is, it has carried the information from the DNA for the construction of amino acids, which ultimately leads to the production of proteins. Okay? Remember, these are the 3 important RNAs involved in protein synthesis. Now, if you look into the machinery, how does it works? You see, this is our DNA. DNA is having 2 strands. Below you can see this will act as a template. And RNA polymerase will be bound over here. So when this RNA polymerase is bound over here, what happens? mRNA synthesis will be occurring, as you can see. Right? Now you got an mRNA. Into this mRNA, your ribosome machinery will be added. Now what you are getting? You are getting a polypeptide chain. Right. So if you see accordingly, we have nucleotides lined up. DNA is made up of what? DNA is made up of your nucleotides. That is 4 different nucleotides are there. And usually replication, transcription, everything proceeds in which direction? 5' to 3' direction. Right. So this acts as a template for the synthesis of the below strand. Now this below strand will be exactly copied in the mRNA. But what could be the difference? Can you identify any difference in those 2 strands? Like the below strand and the mRNA strand, can anyone identify any difference? Can anyone identify any difference over there? As you can see, if you remember, I said you uracil will be placed instead of thymine. So that's why wherever thymine is present, there uracil will be coming. Remaining all the nucleotides will be the same. As you can see, UGG, UUUG. Here also the same thing. GGG, C is coming here. But again T is coming. So instead of that, you are having U. Right. Now this mRNA will be translating to proteins. Now at this junction, what we are having? We are having codons. I will come into codons in details. So codons are what? These are nothing but a combination of 3 nucleotides. So a combination of 3 nucleotides will give rise to a specific amino acid. Like UGG, it codes for tryptophan. UUU, it codes for phenylalanine. GGC, it codes for glycine. So like this, a combination of amino acids will be constructed, and that leads to the production of a polypeptide chain. Now coming to the first important component when we go for protein synthesis, that is mRNA. So what is mRNA? It is a messenger RNA. So there is a small variation when I speak of mRNA with respect to prokaryotes as well as eukaryotes. Do you think prokaryotes and eukaryotes will have the same protein synthesizing machinery, or will it be different? Anyone? Do you have the idea that protein synthesis will be same for prokaryotes and eukaryotes, or will it be different? Anyone? Yes, Pragya, Shivani, anyone? Would like to give the answer over here? Is the protein machinery same or different? Definitely, this is a unicellular and this is a multicellular. So definitely there is variation in the transcription translation. Yes. What is the difference? Because in prokaryotes, we don't have nucleus, right? So the hereditary material will be present where? Will be present in a specified region in the cytoplasm. That's the reason what happens. In that case, we are having coupled transcription and translation. Meaning what? Along with transcription itself, translation will be proceeded. But when we look into eukaryotes, what is the problem with that? Here, mRNA synthesis occurs in a specified location known as nucleus, because our hereditary material, that is DNA, is present in the nucleus. After that, your mRNA will be released into the cytosol, that is specifically speaking the ribosomes, where protein synthesis will be carried out. Okay. So mRNA, I hope so you got the answer, Polaris. So when you go for eukaryotes, monosystronic mRNAs are there. Monosystronic mRNAs means what? There will be one mRNA that codes for one protein. But when you look for prokaryotes, it will be polysystronic. Poly means what? Many. Mono means what? Single. Right. So here you will be having one mRNA that will be coding for more than one protein. So if you look into this picture over here, polysystronic, that means which goes for prokaryotes. P for P. Monosystronic, which goes for eukaryotes. This is considered to be an ORF, open reading frame. That means it is having a start codon, it will be having a stop codon. So corresponding proteins will be synthesized. Here you can see one RBS is mentioned. That RBS is nothing but ribosome binding site. Okay, so that is ribosome binding site over there. As you know, within the mRNA, we are having ribosome binding site, start codon and stop codon. And since this is polysystronic, we are having multiple proteins being generated from here. Right. Now, within this RBS, we have got a specific sequence known as AGG. So, N represents any nucleotide. So, we can consider it as AGG. This is known as Shine-Dalgarno sequence, which is considered to be a conserved sequence. Okay. So, corresponding to this sequence. So, this sequence is present where? This sequence is present in your mRNA. Corresponding to this sequence, we have got another sequence present in 16S rRNA. I hope so you are able to connect why I said you 16S rRNA. See, when we are having protein synthesis, we are having ribosomes. Ribosomes is having large subunit and ribosome is having small subunit. Within the small subunits, you are having certain RNAs. I said you, one of that RNAs is 16S rRNA. Okay. So within the 16S rRNA, you will be having a sequence corresponding to this Shine-Dalgarno sequence. And you call it as anti-Shine Dalgarno sequence. Okay. So when these two are highly complementary to each other, then what happens? Translation rate will be better. There is a significance. Why do you have these two attachments, or why do we have this two complementarity? In case if you have these two complementarity, if these two are very much complement to each other, then only the translation efficiency will be better. Now this I am speaking of polysystronic. But when I speak of monosystronic, you don't have any such sequence. Only you are having a start codon known as AUG, which codes for your methionine. Okay, that's it. So you will be having a single mRNA, and the single mRNA will give rise to a single protein. That's it. I hope so. Polysystronic and monosystronic is clear to all of you? Or is there any doubt in this picture? Clear, right, everyone? So moving ahead, whatever I explained you over here, the same thing. So we have got ribosome binding site. And this ribosome binding site consists of this Shine-Dalgarno sequence. And this Shine-Dalgarno sequence, where it will be present? It will be present in the 5' of the start codon. As you can see, this is your 5', this is your 3'. So towards your 5' only, the Shine-Dalgarno sequence is present. Then this 5' Shine-Dalgarno sequence will be complementary to the 3' end of the 16S ribosomal RNA. That's what I said you. This is your anti-Shine-Dalgarno sequence, which is present within your 16S rRNA. So these two will be complementary to each other. Got it? Next. So these are the sequences I said. AGG, which is your Shine-Dalgarno sequence. CCU and CCA, that is complementary to this one. That's why you call it as anti-Shine-Dalgarno sequence. So if there is high complementarity, there will be active translation. If there is limited complementarity, as I said you, there will be lower levels of translation. Atanu, you could not understand? Which point you could not understand? If you specify, I will explain you again. Then we are having mRNA, right? So this mRNA, as I said you, it contains all the information that comes from DNA. So that information will be in the form of what? That information will be in the form of codons. And these codons are defined as genetic code. Okay, so from mRNA, we are moving to genetic code. Now, what does this genetic code implements? Genetic code implements that it is a combination of 3 nucleotides. So, like you are having UGC, ACG, like that, 3 nucleotides will be there, U, like that. So, these nucleotides will be corresponding to what? Either to a particular amino acid or to a stop signal. If you remember, I had shown you in this picture, right? You are having UGG, which acts as a single codon, and this codes for what? Tryptophan. UUU. This is a single codon, and it codes for what? Phenylalanine. So like that, glycine, serine. So for all the 20 amino acids, you are having the codons. So you might be thinking we are having 20 amino acids. But to code for this 20 amino acids, we are having a total of 64 codons. So for one amino acid, you need 3 codons, like 3 nucleotides, right? So you can imagine why 20 amino acids needs this 64 codons. The reason for this is, I'll come to that point. For understanding why do we need 64 codons for 20 amino acid, we must understand the properties of genetic code. Now, what are the properties of genetic code? Our genetic code is a triplet codon. That means it is consisting of 3 successive nitrogenous bases. And our code is comma-less. That means if AAG, if this is producing one amino acid, there is no comma in between. Rather, simultaneously, there will be another amino acid like this UAC or something like that will keep on moving ahead. The code is non-ambiguous. Non-ambiguous means a particular codon will always code for the same amino acid. Meaning what? Suppose UUU, it codes for phenylalanine. So this UUU will always code for phenylalanine, not for anything else. But for everything, there are exceptional cases. Here also we are having the exceptional cases. As you can see, AUG is acting as a start codon for almost all translation mechanism occurring in eukaryotes. But in some cases, GUG can also act as a start codon. In fact, GUG is coding for valine as well. So this indicates what? There are some exceptional cases when we go for genetic code as well. Another example, GGA. This is one codon which codes for two different amino acids, glycine and glutamic acid. So depending upon the situation, the glycine or glutamic acid, they will come into play. Next, what you are having, the code is universal. Universal means what? 64 Atano, 64. Okay. The code is universal means what? Same genetic code is found valid for all organisms ranging from bacteria to man. That means what? UUU. If it is phenylalanine for me, it is phenylalanine for you as well, and it is phenylalanine for bacteria as well. For all the organisms, I'm saying. So that's why you call it as a universal. And the last important property of a genetic code is degenerative. That is, our code is degenerate by nature. Meaning what? Excepting your methionine and tryptophan, you can see they are having only one codons each. But remaining amino acids, you see, asparagine is having two codons. Isoleucine is having three codons. Alanine glycine is having 4 codons and leucine arginine is having 6 codons. So one amino acid can be coded by more than one codons. So that is known as degeneracy. Okay, so I hope so genetic code is clear to all of you. If genetic code is clear, mRNA is clear. Now, if after mRNA, the second important component when it comes to protein synthesizing machinery is your tRNA. Now, what is the function of tRNA? Amino acids that are being produced by your mRNA has to be carried. And this carrier, or you can say the adapter, is your tRNA. So, in order to attach tRNA to your mRNA, you must have some sequence complementarity, right? Then only they will be attached with each other. For mRNA, you know they are having codons. Corresponding to that, for tRNAs, they are having anticodons. Okay, so anticodon will be complementary to the codons. And this code will determine the type of amino acids and the order in which they are joined will be specifying a protein. That means what? Suppose insulin has to be synthesized, glucagon has to be synthesized. Do you think the amino acid construction will be the same for both the cases? Do you think the amino acid construction is same for both the cases? For constructing a protein like insulin and glucagon? No. Right. The amino acid composition will be different. That is what it says. It determines the type of amino acid which specifies that protein. And the sequence in which the amino acids are added will determine its structure as well as its function. Okay. So, remember this important thing. Codon is for mRNA, whereas anticodon is for tRNA. Remember, this is important when you go for translation. You should not exchange. Codon for mRNA, anti for tRNA. So, you can remember anti, TI, tRNA. Remember this code word. Next, what we are looking into? We are looking into the structure of tRNA. So, among all the RNAs, tRNA is the smallest one. its secondary structure is called clover leaf structure, and its tertiary leaf structure is called inverted L-shaped structure. And this tertiary structure is being stabilized by your hydrogen bonding, base stacking, as well as hydrophobic interactions. If you look into the structure of this tRNA, you can see that this is your anticodon loop and this is having the anticodon. This is anticodon of tRNA, codon of mRNA. So these two are complementary to each other. Unless and until these two are complementary to each other, your tRNA and mRNA will not interact. And if there is no interaction between tRNA and mRNA, then there won't be any protein synthesis. That's why complementarity is very important. Then, I have said that tRNA is responsible for carrying what? Amino acids in order to create a polypeptide chain. So, these amino acids are carried in this 3'. See, this is your amino acid attachment site. Okay. So, this becomes your tRNA. Now, you understood mRNA. What is an mRNA? You understood what is a tRNA. Now you have to understand how tRNA is getting activated, or I can say how you are charging the tRNA, right? For charging the tRNA, as I said you, if you remember, I said you or I had mentioned you about amino acyl tRNA synthetase. This is the enzyme I mentioned, the third category. So this enzyme is responsible for what? This enzyme is responsible for the activation of tRNA to carry out protein synthesis. Now, how they are going to activate your tRNA? Suppose we are taking amino acid valine, right? So this enzyme, into this enzyme, valine along with ATP, that is adenosine triphosphate. You can see 3 phosphates are also moving along with adenine. So these two are moving and attached to the particular enzyme. Which enzyme? Amino acyl tRNA synthetase enzyme. Always remember, any enzyme that ends with thetase, specifically synthetase, will take the energy from ATP or GTP. If you look out any kind of metabolic reactions, metabolism chapter if you have, or if you are going to study in the higher classes, there you will come across that these kind of enzyme seeks the help of either ATP or GTP for their activation. So what happens? Valine goes and valine along with ATP goes and binds to the enzyme. Due to this binding, what happens? ATP underwent hydrolysis, which releases 2-phosphate outside. So what you are retaining? You are retaining adenosine monophosphate with amino acid along with the enzyme. Okay, so these are the 3 components. Now, into this, you are going to add an uncharged tRNA. So this uncharged tRNA will be approaching the enzyme. So at that time, what happens? The corresponding enzyme, which enzyme? Your amino acyl tRNA synthetase enzyme, what it will do? It will transfer this valine into this 3' end of tRNA, as you can see in this picture, the last picture, and your AMP is released. So now what you got? Amino acid tRNA enzyme. The enzyme will be releasing the activated form of your tRNA. And this is how the amino acid is attached to the tRNA, which will be taken to attach or to find complementarity with that of mRNA. And this is a very important step when we go for translation. Charging of tRNA. Charging of tRNA with correct amino acid. I hope so this picture is clear to all of you. Any doubt in this picture? No, right? You are able to understand? Remember, this is important when you go for any exams related to this topic. Next, what we have to say, amino acyl tRNA will be delivering the amino acid to the ribosomes. Why it is delivering to the ribosomes? Because ribosomes are the ultimatum for protein synthesis. Okay. So when I say a translation complex, how to define a translation complex? It is composed of three important ingredients. What are those three ingredients? Ribosome, mRNA, and charged Trna. So when these three components combine together, you are starting a translation event. Okay. So next what we are having, we are looking into the structure of ribosomes. You know that ribosomes are made up of two subunits, larger subunit and smaller subunit. So this becomes your mRNA and this becomes your tRNA. Okay. And within this larger subunit, you are having three sites known as A site, P site and E site. What is the significance of these three sites with respect to translation? You will be coming to know within few slides. So just remember, ribosome is composed of two different subunits. And it is in this P site you will find peptide bonds are formed. Do you have any idea how peptide bonds are formed? Anyone? Yes, good evening, Nilanjana. Any idea how peptide bonds are formed? Anyone having any idea how peptide bonds are formed? No? I will come to that point as well shortly. So, prokaryotic ribosome, you know that already prokaryotes and eukaryotes are different when it comes to translation, right? So, when you look into prokaryotic ribosome, it is made up of a large subunit and a small subunit. That is known as 50S subunit and this is 30S subunit. So, I have already mentioned you that this ribosomes is composed of what? rRNAs. And I have already given you the significance of 16S rRNA. Do you remember the significance of 16S rRNA? I mentioned in the initial few slides. Anyone remembers? Nilanjana, Central University Entrance Test. Anyone remembers the significance of 16S rRNA? It is having your anti-Shine Dalgarno sequence, which will go and bind to the Shine Dalgarno sequence, which is present within your mRNA in order to initiate the translation. Right. So that is what I mentioned if you remember. So combine these two subunits, you are getting a 70S subunit. So 70S subunit is your prokaryotic ribosome. Similarly, if you look into the eukaryotic ribosomes, the division will be 60S subunit and 40S subunit. And accordingly, the rRNAs will also be different. As you can see, here in your 60S subunit, you will be having 5S, 5.8S as well as 28S RNA. But when you look into 40S subunit, you will be having 18S RNA. So different subunits will be having different sets of ribosomal RNAs. And all of these RNAs will have some specified role and it will undergo transcription with the help of RNA polymerases. Meaning different types of RNA polymerase. Actually, our RNA polymerase is divided into 1, 2 and 3. Right. So depending upon the RNAs, the polymerases will be transcribing. Next, what we are having, I said you about the three sites. That is your A site, P site and E site. Where it is located? These are located within your large subunit. Large subunit of what? Large subunit of our ribosomes. So what is the significance of these? Are actually gates you can see. Okay. Or you can say sites. Anything, whatever you wish. So A site corresponds to what? Amino acylated tRNA. I hope so you remember how the tRNA got amino acetylated. That means charging. Okay. So this is the location for your charged tRNA. Okay. Next, what you are having? P site. P site corresponds to what? Peptidyl tRNA. Always remember one thing. You have got initiator tRNA, right? Initiator tRNA is present. It will be carrying which amino acid? Which amino acid is being carried by your initiator tRNA? Any idea? AUG codes for what? Which amino acid? AUG codes for which amino acid? It codes for methionine, right? So the first tRNA will be occupying the space in this P site. Remember this one. First tRNA. Whereas upcoming tRNAs will be occupying the space in your A site. Yes, very correct, Polaris. Then you will be having the exit site. What is the significance of exit site? From here, your old tRNAs will be released out. That means after the peptide bond formation, whatever the existing tRNAs are there, they will be released out. From where? From this E site. Remember this point. Okay. I hope so. The significance of E site, P site and A site is clear to all of you. See, here it is written. E site is the binding site for tRNA that is being released after the growing polypeptide has been transferred to your amino acyl tRNA. Means here you are having a tRNA that is carrying an amino acid. This amino acid will be forming peptide bond with this amino acid. After that, what happens? The tRNA will give this amino acid to this amino acylated tRNA and this tRNA will be released out. So once this P site tRNA is released out, your A site tRNA will occupy the space of P site. And into your A site, new tRNA will be added. Okay. So what do you think? Is mRNA is moving? Is tRNA is moving? Or is ribosome is moving? Out of three components, which component might be moving when we go for translation? You have to scan it, right? You have to get an AUG, that is start codon, then only you should start translation. So who is scanning this AUG? Is it our AUG is present in mRNA? So will it be your ribosomes or will it be your tRNA? Who will be scanning? Yes, it is your ribosomes that are responsible for scanning your initiation codon. After that, only your tRNA will come and bind over there. Okay. So when you look into the steps of translation, we are having initiation, elongation, and termination. These are the three basic steps that are involved in translation. Even if you go for replication, transcription, there also you are having like the steps. Initiation is there, termination is there. Okay. So first is the initiation. Now, what does initiation will do? The main purpose of initiation is to scan for AUG, that is your start codon. Okay, and who is responsible for the scanning? The small subunit of your ribosome. The small subunit of ribosome is responsible for scanning this AUG. So when I speak of prokaryotes, you are having specific sequence, and when it comes to eukaryotes, you don't have any specific sequence. Right. Next, small subunit will move down scanning until and unless it encounters your AUG. Right? And to encounter this AUG, they must have some initiation factors involved. So for prokaryotes, you are having initiation factor 2. For eukaryotes, you are having E for eukaryotic, initiation factor 2. And to recruit this initiation factors, you need energy. That energy will be provided by your GTP. Remember this point. Okay. So, in fact, when you go for prokaryotes, 3 initiation factors are there. But when you go for eukaryotes, more than 3, you can say large number of initiation factors are there. Okay. So as I said your initiated tRNA will find its location where? Within the P site of your small ribosomal subunit. Remember, E is for exit. P is for initiator tRNA and A is for acylated tRNA. That is your new tRNA. Right. again, there is a small distinction when it comes to prokaryotes and eukaryotes. Your initiated tRNA will be having a formyl group attached to methionine when you go for prokaryotes. But when you go for eukaryotes, you'll be having only methionine. So after your mRNA comes, your tRNA comes, your small subunit comes. When all this has accommodated properly, then only the large subunit of your ribosomes will be joining that particular complex. So if you see, here I have provided you a table. Of course, currently you might not be requiring this much details. When you go for higher and higher studies, you will be requiring in each and every functions of this initiation factors. Right now, you can remember initiation factors are required in order to initiate the translation process. So when it comes to prokaryotes, as I said, you three factors are there. And among this, it is your initiation factor 2 which requires the energy from GTP. But when you look for eukaryotic factors, you can find numerous number of factors are there. And here also you are having only your eukaryotic elongation initiation factor 2 which requires GTP hydrolysis. So as I said you, when you go for higher studies, you will come to know about details about all these things. Next what you are having? Elongation step. Elongation means what? Where you are making the peptide bond, right? Any idea how peptide bonds are created? Any idea how peptide bonds are created? How the two amino acids are joined together resulting in peptide bond formation? Any idea? Anyone? Pulkit, Nilanjana, Atanu, Polaris, anyone having the idea? See how the peptide bonds are formed. This is your first amino acid. It could be any amino acid, okay? That is having an N terminal, C terminal and having an R group. This is your second amino acid, N terminal, C terminal and an R group, right? These two amino acids has to be combined together. How they will be combined? With the help of peptide bond. How these peptide bonds are formed? See, when these two amino acids are combined together, this OH and H will be released as water. And whatever the remaining content you will be seeing over here, they will be joined together forming a peptide bond. So this is how a peptide bond is constructed among the amino acid. And this is being carried out during the elongation phase. As you can see, it requires an enzyme also for that, that is peptidyl transferase enzyme that helps in this bond formation. So the amino acid that was attached to tRNA at P site, who was attached at the P site? Your initiator tRNA. Now they will be linked to where? Linked to the amino acid that is present in the A site. If you remember, I said you from A, from P and E, whatever the amino acid will be here, they will form peptide bond over here. So what happens? The tRNA which is present over here will be exiting the ribosomes. And from here, the tRNA will be occupying the P site. Now, this site will be occupied by a new tRNA. If you remember, I had said you previously. Then what happens? After that, translocation occurs. That's what I said you. Translocation. From here, the tRNA will be moving to here. And from here, the tRNA will be released out. And this space will be occupied by new tRNAs. Okay. So, what is the next thing? So, like initiation factors, when we go for elongation, even you are having a large number of elongation factors as well, both for prokaryotes as well as eukaryotes, which is carrying out specified functions under the elongation process. Again, when you go for higher studies, you will come to know about all these things in details. But right now, you remember elongation factors are involved in the process of peptide bond formation. Next, the third step is what? Third step is termination. So, we know that we are having one start codon, that is AUG, and three stop codons. right? And the stop codons is otherwise known as nonsense codons. Nonsense codons means what? That does not make any sense. That means it does not code for any amino acid. So accepting this three, remaining codons will be considered as sense codons because they are making sense, means they are able to code for specific amino acids, right? Now what happens? Release factors will be joining. So when the process has to undergo termination, we have got a group of proteins known as release factors. They will be moving towards the A site. At that junction, what happens? Polypeptide will undergo hydrolysis. Again, when it comes to prokaryotes, you are having release factor 1, release factor 2. And when it comes to eukaryotes, you are having only release factor 1, 2, and 3 is also there. I will show you in the end of the, within the session. So after that, what happens? Your polypeptide chain is released and this polypeptide chain will undergo modification. Why they are undergoing modification? So that they can go for transport. Right. So what is the fate of mRNA, tRNA, ribosomes? All those things will be recycled and can be utilized again. Right. So that comes your termination. So if you look into this picture, this comprises of three steps. First is initiation, second is elongation, and third is translocation. Okay. In the initiation, as you can see, this is your A site, this is your P site, and this is your E site. As I said you, it is in your P site, you will be having methionine present, initiator tRNA present. And this is the complementarity. AUG initiator codon complementary to the tRNA in order to initiate the translation. And in this site, what you will be having, you will be finding new tRNAs. As you can see here, new tRNAs are formed. So new tRNAs are formed and amino acids are formed like this. So these amino acids are connected by what? Peptide bonds. So after some time, what happens? This tRNA, which is having your methionine, will be transferred to this amino acid tRNA. And after that, it will be exiting from here. So this will occupy this space and another tRNA will be occupying this space. That process is known as translocation. Okay, as you can see, ribosome translocates in the 5' to 3' direction. tRNA in the P site will be entering the E site, that will be exiting. And your A site will be occupying a new amino acid tRNA. So this is one picture. In case if you are not able to understand here, there is another picture for you. See. These are the components. You are having the tRNA which carries the amino acid. This is the ribosome with large and small subunit. And this is your stop codon which is necessary to undergo termination. Right. So upon initiation, what happens? Your initiated tRNA will come and bind to the P site. After that, amino acids will be added. And this amino acid, as you can see the arrow over here, the amino acids will be transferred to this tRNA. And this tRNA will be released outside. Okay. And from here, you can see release factors. So that becomes your elongation. That is the bond formation. So from here, the amino acids will be joined to this amino acid tRNA and that will be released. Another amino acid tRNA comes into play. When you go for termination, what happens? When the ribosome is keep on scanning the mRNA, it reaches a specified stop codon. It could be any of the three stop codons we have seen. Suppose if it is UAG. So when this ribosome encounters the stop codon, what happens? Your release factor will be recruited to the A site. So when this release factor is recruited to the A site, at that time what happens? There undergoes polypeptide hydrolysis. As you can see here, the polypeptides will be released from the tRNA. Now this polypeptide will undergo folding and this would lead to what? This would lead to protein synthesis. So, did you understand translation? Everything clearly? How the process is being carried out, how the factors are being involved, how the translocation is occurring, how the peptide bond formation is occurring. All these things are clear to all of you? Yes? So this is a kind of summary which I can give you with respect to the factors that are involved in your synthesis, that is protein synthesis. As you can see, you are having initiation factors, elongation factors, and release factors. If you see, as I said you, if you remember, RF1, RF2, RF3 is present. So RF1 is specific for identification of UAA and UAG. But if you go for RF2, they are specifically recognizing UAA or UGA. These three are codons, termination codons. And RF3, they are responsible for stimulating other release factors. Other release factors means your RF1 and RF2 only. Okay. So this completes the basic idea. And this is just a comparison of translation when I say of bacteria and eukaryotes. The major difference among these two translation machinery is the ribosome. This is 70S ribosome and this is your 80S ribosome. And in this case, FMET is used as an initiator tRNA, and in this case, methionine is used as an initiator tRNA. Shine-Dalgarno sequence, which ensures complementarity and stable binding between your tRNA and mRNA. It is present in bacteria, but it is absent in eukaryotes. Simultaneous transcription and transcription, that is, at the time of transcription itself, translation is occurring in case of bacteria. Why? Because of absence of nucleus. But in this case, it is not possible because of presence of nucleus. So within the nucleus, mRNA will be synthesized. That will move out of the nucleus, enters the cytosol. There it will come across ribosome. And within the ribosome, you will find protein synthesis occurring. Right? So this is a comparison table. And these are some antibiotics, or you can say toxin, who can target either prokaryotic cells or prokaryotic and eukaryotic, or maybe eukaryotic cells. Just for an example, I am saying tetracycline. This is an antibiotic whose target is prokaryotic cell. So, if this tetracycline, the target will be the A site of 30S ribosomal subunit. So, just imagine if this tetracycline goes and binds to the A site of 30S ribosomal subunit, what does it leads to? It leads to inhibition of amino acyl tRNA binding to the A site. Right. Next, if you see puromycin, it is present in both pro and eukaryotes. In this case, what happens? They are targeting the peptidyl transferase center. That means your P center is being targeted. That means what? P, that is your peptide bond, will be terminated. If you see diphtheria toxin, they will be targeting your eukaryotic cells. And they are responsible for modifying the elongation factors. So, elongation will be inhibited. And the last one, if you see cyclohexamide. If you see cyclohexamide and chloramphenicol, their mode of action is same. That is, they are targeting the peptide center. But their target is prokaryote and here it is eukaryotes. Function remains the same. Target will be varied. Okay, so these are the antibiotics. Now, giving you a mnemonic for translation. That means if you find anywhere confusion from where mRNA is coming, where it goes and binds, for all those things, in order to clarify it completely, you can remember this mnemonic, Mr. Cat App. Now, what is this Mr. Cat App represents? C. M stands for messenger RNA. So this messenger RNA will go and bind to the ribosomes, right? So this ribosomes will read the sequence in codons, that is for C. And these codons are recognized by whom? Anticodons, as you can see here. And these anticodons are found where? They are found in transfer RNA. So this transfer RNA carries what? Amino acids. Now, this amino acids, if they are joined together, they form peptide bonds. peptide bonds amino acid combination will lead to the production of polypeptide chain. So you can understand, you can go through this mnemonic even during the exam time. If you feel any confusion in any of the step, you can remember this particular word. Okay. So before signing off, just wanted to remind you, if you have any form of technical queries, you can drop a mail at info@biotecnica.org, or you may contact to our toll-free number 1800-1200-1818 or 080-5099-700. And you are most welcome with the feedback. You can drop your testimonials in this zipmessage.com. Last but not the least, thank you for your patience for staying till the end. Before winding up, wanted to say, miracles start to happen when you give as much energy to your dreams as you do to your fears, right? We are having the exam. What is our common human mentality is that we become afraid. We become scared. Acha, tomorrow is the exam, so am I able to recollect the things? All those lot of questions pop up in our memory. So that is a kind of energy drain out. But if you put the same energy into your exam, for working towards your exam, working towards your dreams, you can see how much beneficial it will be, right? Okay, then thank you so much, my dear students. Wishing you all the best for the upcoming exam. And please join back tomorrow. Tomorrow also you are having a session from the same unit, genetics and evolution. Thank you.